Hello, everybody. This is Todd Sherer from the Michael J. Fox Foundation, and I want to thank everybody for joining the sixth of our Hot Topics calls this year, and this is our year-end Parkinson's disease research year in review, 2012. And the topic for today that I'd like to cover is really both looking back and looking forward in terms of Parkinson's disease drug development and getting a sense for how far we've come and also what lies ahead going forward. We've actually had a very exciting year in Parkinson's research with a lot of new data, new results, but obviously there's more work to be done. So just to review the agenda for today, we've already completed the welcome, and I will just do a quick recap of how we get to cures, what is the process, where are the challenges, and what can be done to try to accelerate and overcome some of those challenges. And then we wanted to take a step back and just look at where was Parkinson's disease research in 2012, and now where are we 12 years later? Where has it advanced? What avenues have not shown as much promise? Really focusing on some specific highlights from 2012 and what to look forward to in 2013 and beyond. And then we'll have plenty of time for question and answers, both with myself and with our on-staff neurologist, Maurizio Facheris. So let me just jump right in now. So this is just to recap, how do we get to cures? How do we convert a basic discovery, a new piece of information that could be related to Parkinson's disease, move it through all the stages up through clinical testing and ultimately regulatory approval? This is a very expensive and risky process and really depends on a lot of players getting involved, researchers, pharmaceutical industry, and an important role that the patient community can play. So rather than me try to give a detailed presentation on each of these sections of the development pipeline, we actually have developed animation video that I think really tells this story in a very, very compelling way. So I'm going to next turn over and play that video. It does have sound, so if you're watching it, just make sure your audio is on your computer. So let me just go to the video now, and then we'll move on with the discussion. So, okay.
Hi, I'm Debbie Brooks, the co-founder of the Michael J. Fox Foundation for Parkinson's Research. We launched in 2000 with the goal of delivering better Parkinson's drugs to patients and ultimately finding a cure. So we've had a few years to learn just how complex drug development is. Sadly, to bring one new central nervous system drug to market takes 15 to 30 years and well over $1 billion. So what does it take to get an aha moment to the drugstore shelf? Just explaining drug development is a challenge, so I often use the alphabet as an analogy to help make sense of these complicated concepts. The first part of the alphabet, say letters A to F, represents basic research. This is where a scientist, typically an academic, looks at some molecular process in our cells and asks, what's happening here? Why is it important? As taxpayers, we fund over $30 billion a year at this stage through the National Institutes of Health. This discovery science is the backbone of drug development worldwide, but aha moments alone are not enough to make a difference in human health. The next chunk of the alphabet, say letters G to P, represents translational research. Discoveries worked on for years in basic science are now examined in the context of a particular disease. This work helps us identify ideas that may actually get us closer to new medicine and enable the leap of faith from testing in a petri dish to testing in a human being. The bad news is that most ideas will fail at this stage. It's the valley of death where breakthroughs go to die. The very few therapies that do make it through the valley of death still have to navigate Q to Z, clinical research, where potential new drugs enter human testing. Clinical research is a lengthy, multi-phase process, and each phase requires years and new rounds of funding. At this stage, it's necessary to design trials, recruit volunteers, collect data from patients, analyze the findings, and finally, prep for approval from regulators before a drug can become part of a physician's arsenal. Across all diseases, the total yearly expenditure here is about $70 billion, mostly in private investment from drug companies. But even after getting as far as Q to Z, only 1 in 12 of those treatment ideas are ultimately proven safe and beneficial to patients. Between government and industry capital across that whole alphabet, we're up to $100 billion a year spent on drug discovery and development, and we have shockingly little to show for it. The Fox Foundation's total research investment to date measures in the hundreds of millions. That may sound like just a drop in the bucket given billions are required, but we believe getting results for patients isn't merely about spending more money, it's about spending the money smarter. That's why 90% of our funding goes to moving ideas into that middle part of the alphabet and beyond through the valley of death to the clinic. Our strategic investments identify and chaperone the ideas that are most likely to have an impact on human health. We provide researchers with the financial and intellectual resources they need so that they can get these results as quickly as possible. Michael J. Fox has a great way of thinking about our foundation's role when he talks about a department of cures. We'd all like to imagine an office where a high-level official sits at the center of all this work with a smart and comprehensive view of the entire field, orchestrating the efforts of all the players working on these complex problems. And then we'd like to have someone whose job it would be just to keep the whole business moving in an efficient way to assure that the best ideas are on a streamlined path towards the patients who need them. Sadly, there is no department like that. But this is the role our foundation is endeavoring to play in Parkinson's disease research. We've built a team of PhD neuroscientists paired with business-trained project leaders. Sitting at the global nexus of Parkinson's research, this team comes to work every day with one urgent goal: to prioritize research that can transform patients' lives. And in our short history, we have built the credibility and expertise to make real inroads. We understand not only science, but the business of science. And we're willing to take higher risks that can tilt the odds in favor of success. And we are seeing real progress. Today, more ideas are flowing into translation, and many of them are progressing from there to that vital Q to Z phase. Our work goes far beyond check writing. We really assess what is needed to get from A to Z, and then we bring it. I wouldn't want to leave you with the impression that any of this is easy. We must be sober about the work that needs to be done. But it'll be a great day when we find that last breakthrough. Patients have the treatments they need, and we can close our doors. Until then, we will keep at it and do whatever it takes. We're problem solvers, and we're optimistic.
Okay, so I think that video provides a good understanding of all the steps in drug development and the challenge that we're facing, but different approaches that could help us overcome those challenges. Anyone that had problems viewing that video, we do have it posted on our website, and we will send out a link so you can read it. So hopefully it worked. This was our first time trying a video in this, so hopefully it worked. And I do think it's a good way of telling the story. What I wanted to jump into now was just taking a step back and looking at where were we with Parkinson's disease research in 2000. And I think it's safe to say that at that time, Parkinson's disease research was at a bit of a crossroads. A lot of work had been done on developing dopamine-based treatment options. There was a lot of dyskinesia seen in patients in response to some of those treatments. Deep brain stimulation was just coming on board, just recently approved. But really, there wasn't a lot of innovation in the works around symptomatic treatment, as most of the work focused on improvements in dopamine-based therapies. So dopamine, again, is the neurotransmitter or the chemical in the brain that is lost in the disease that controls movement. Most treatments involve replacing that chemical, replacing that dopamine. And that was really a lot of the state of the field at that point in 2000, and still is actually a very effective treatment for many symptoms of the disease. But in 2000, there was a very little appreciation of non-motor symptoms of Parkinson's. Not much research in those areas, things like sleep or depression or even some cognitive issues that can be part of the disease. And there was not a lot of research beyond dopamine in terms of trying to develop symptomatic treatments. For disease-modifying treatments, treatments that can actually target the underlying disease process, there really was not a lot of novel activity going on. We had a very limited understanding of the underlying disease causality and underlying disease biology. But there were some active efforts. Stem cell therapy was very exciting, controversial. There were ongoing transplant studies in Parkinson's. We also had some early clinical evaluations of neurotrophic factors, which are proteins that can bring brain cells back to function better, sort of like a fertilizer for the brain to get that brain functioning better. And those were in early testing. One of the real exciting things that had just come on board in 2000 was the emerging role of genetics. So the first genes associated with Parkinson's disease had been discovered. They were very poorly understood, but they really pulled the envelope on a complete new direction of research that has now been explored for the last 12 years. And the field had a number of systemic challenges. There was no centralized player really coordinating the field-wide approach. This was addressed in the video. And an honest assessment was that the industry, pharmaceutical industry interest in Parkinson's disease was dwindling, particularly because it was felt they had played out the dopamine-based therapies and there wasn't a lot of biology pointing to new directions. So just now going forward to just give a more schematic of where we were specifically in 2000. In terms of disease-modifying therapies, things like alpha-synuclein, the new genetic targets, were in their infancy in Parkinson's. Trophic factors had just reached clinical testing, and it was showing some early promise. And a lot of the new ideas that we now are more familiar with in Parkinson's, things like calcium channels as therapeutic targets or antioxidants like urate, were really just coming on board. In terms of symptomatic treatments, there really wasn't a lot of very exciting new targets being explored. Dopamine therapy had really been developed. Deep brain stimulation was just coming on board. And we were just really starting some of the new approaches focused on other brain chemicals that I'll talk about more now as we get into what we've learned since 2000, now in 2012. So this is now the same outline, but really looking at where are we now in 2012. And what you can see is that the genetic targets like alpha-synuclein are progressing. Just this year, we launched the first clinical trial targeting alpha-synuclein. A new gene has come on board through discoveries called LRRK2, which is the most common genetic cause of Parkinson's disease, which is now reaching later stage preclinical development, moving towards the clinic. And we've really progressed some of these other targets. Trophic factors are now in phase 2 clinical testing, the second phase of clinical testing. And these other targets, calcium channels, antioxidants like urate, these are now in clinical evaluation. And you can also see now a much more robust pipeline when we look at the symptomatic targets. There are therapies in the clinic for cognition, therapies targeting other brain chemicals like glutamate and serotonin focused on dyskinesia or psychosis that are in later stage development. There's a lot of very interesting work ongoing to improve levodopa therapy that is in clinical testing. And another target, and I'll talk about this in more detail, adenosine, which is another brain chemical, is in late-stage development for motor symptoms, with even recent results just announced this week. So with improved knowledge about the disease, an improved understanding of the full spectrum of the disease, and improvements in our ability to prioritize targets, we've really seen pretty significant progress when you compare the landscape in 2000 to the landscape in 2012. Just what are some of the critical developments that have helped to drive this progress? And Some of these really focus in the area of disease-modifying trials. There's been a lot of focus on developing markers, biomarkers, objective measures of the disease to improve our diagnosis, as well as to improve our ability to track the disease, to really determine whether a therapy is altering that disease process and the course of the disease. Just earlier, I think it was either last year or earlier this year, DAT scan approved in the United States, which is now FDA approved for monitoring the levels of dopamine in the brain, which helps with the diagnosis of Parkinson's in combination with the clinical motor evaluation. A number of major initiatives are now ongoing and starting to develop data around biomarkers, including the Fox Foundation Parkinson's Progression Markers Initiative. And there are very, very specific efforts around the genetic targets of alpha-synuclein and LRRK2 to try to develop the tools to really enable the clinical testing. And one important point I wanted to make about these genetic targets is that with increasing understanding in biology, it's clear that these same proteins, these same targets, play a role in a very large-scale way across Parkinson's disease, not just in individuals that may have the mutation. So, for example, with alpha-synuclein, some individuals have a mutation in alpha-synuclein that can cause Parkinson's, but in every person who has Parkinson's disease, there is an abnormality, an alteration in the function of alpha-synuclein. So this is how a genetic target has now led to the potential for a widespread advance. And in terms of symptomatic trials, there's a lot better clarity now on how to actually go about doing clinical trials to test some of the side effects of Parkinson's treatment, like dyskinesia, and also some of the non-motor symptoms, like cognition, like anxiety, that have had a lot of advances in terms of our measurement and ability to really determine whether a therapy is having a beneficial effect. The other thing that we've seen over the last 10 or 12 years is a complete change in engagement across all the stakeholder groups in Parkinson's disease that is resulting in driving faster progress. This has to do with novel models in terms of how researchers are collaborating both across academic and industry partnerships. Industry has taken a much more open view towards innovation and how to go about interacting with academics and foundations like the Fox Foundation in order to develop pre-competitive efforts to really move progress faster, more sharing of data, more sharing of research experiences and tools. There's a much greater industry involvement in Parkinson's disease now, focused a lot on disease-modifying treatments and building off the discoveries in genetics, but also an appreciation of the non-motor symptoms and some of the side effects. So we'll talk about some of the investments in progress in those areas. But I think probably most significantly is the greater engagement of the patient community in not only demanding improvements in therapy, but also driving that progress by getting involved in clinical trials and efforts like the Fox Trial Finder initiative. Now I just want to go into some specifics of the highlights of 2012, and these are all results and advancements that happened just this year. And I've been at the Fox Foundation for 8 years and have been in Parkinson's research for 15 years. And it's really, in taking that perspective, very exciting to see all of these positive results in the same year happening across the spectrum of Parkinson's. So it's a sort of a culmination, I think, of a lot of work over the last 5 to 10 years, with the major caveat that there is still a lot of work to be built on from these results in this year. But we received this year positive clinical trial results in symptomatic treatments, and all of these that I'm about to go over going beyond dopamine in terms of their therapeutic target. So this is targeting brain chemicals that are affected in Parkinson's besides dopamine, which means that these therapies have the potential of working in combination with the dopamine therapies as an additive therapy, overcoming some of the side effects of current treatment, and also addressing some of the symptoms that are not currently improved by dopamine treatment. So one area that has seen a lot of advances is in glutamate treatments for Parkinson's, and I know some questions were submitted actually in advance on glutamate treatments for Parkinson's. Two studies, one by Novartis, another by a biotech company, ADX, and as a disclosure, the Fox Foundation funded partly the ADX study. But both of these showed that by targeting the glutamate receptor, the glutamate system in the brain, you could reduce dyskinesias, which are side effects of the levodopa treatment. Both of these studies were at the phase 2 stage of clinical development, so there's still phase 3 trials that will need to be done. But it's very exciting that two independent studies showed similar results targeting the same system in the brain. Just this fall, we also received news of a project led by Acadia, which is a biotech company in San Diego, showing positive results. This one was at the phase 3 stage of targeting the serotonin receptor to treat psychosis in Parkinson's disease, which is both part of Parkinson's disease but also a side effect of some of the symptoms, things like hallucination, visual hallucinations. So this project is showing a lot of promise. And we just got some results this week looking at adenosine, which is another brain chemical, as a treatment for the motor symptoms of Parkinson's, as an additive treatment to the dopamine-based therapies. This was a phase 2 trial that was announced by a company called Biotai, a recent earlier clinical study by a company called Vernalis. And we also know that Merck, which is the large U.S. pharmaceutical company, is currently doing a number of large-scale trials developing an adenosine-based therapy. So this is all to say that we now have a robust late clinical pipeline of non-dopamine-based symptomatic treatments for Parkinson's. So that's very exciting, and that all happened in 2012. I hope I have equally exciting news to share in 2013. Also, just in terms of the symptomatic treatment and going beyond dopamine, a significant collaboration was announced between researchers at Vanderbilt University and Bristol Myers Squibb to develop additional glutamate treatments for motor symptoms of Parkinson's that's moving towards the clinic. This is a very exciting partnership that involves Vanderbilt, the Fox Foundation, and now the project has the support of a large pharmaceutical company with the resources that we need to move it through clinical development, so that model that was discussed in that whiteboard video. There also were some novel clinical trials launched in 2012. I mentioned that with Fox funding, a company, Facheris, has launched the first clinical trial ever to target alpha-synuclein in the brain of Parkinson's patients using a vaccine approach. And we should have results from that trial probably next year. And a company called Civitas Therapeutics, which is a Boston-based biotech company, has launched a trial that is developing an inhaled form of levodopa to limit on-off periods. That's showing some early promise. So that's exciting that we continue to fill the pipeline. Just some other highlights that from 2012. We continue progress in the quest for the biomarkers of Parkinson's. The Parkinson's Progression Markers Initiative has been releasing the first results, showing some promising both blood and spinal fluid-based markers. We actually also have to keep a close eye on the Alzheimer's field. There were a number of large-scale trials in the Alzheimer's field, one that did not show positive results. A recent one by Eli Lilly that showed a hint of some positive results, where they're now going on to do additional trials. The reason that we need to keep a close eye on this is really for two reasons. One is to make sure we learn as much as we can from a biology and clinical science perspective, given some overlap of underlying biology between the two diseases. So there's a lot we can learn from their experience. And second, in a lot of these pharmaceutical companies, similar departments manage their Alzheimer's and Parkinson's portfolios. So the funding for the research is very much linked, and if there are successes in Alzheimer's, it certainly will lead to greater investment in Parkinson's disease. So that's something we watch very closely at the foundation to make sure we can maximize the leverage of what's going on in other diseases like Alzheimer's. Some other highlights from this year: the Nupro patch was brought back on the market in the U.S. We also know that the levodopa carbidopa intestinal gel product, which is available in Europe, is now approaching review in the U.S. This is a product that directly infuses the levodopa into the intestine to improve the delivery of the drug. And finally, a treatment for orthostatic hypotension that's being developed by Chelsea Therapeutics is currently under FDA review for Parkinson's disease. So hopefully we'll have information on that coming up soon. So I wanted to give a sense of the exciting progress in 2012, and I think there was a lot of it, obviously a lot to build on and work to be done in 2013. What are some other things to look for in 2013? In spring, we are expecting the results of a phase 2 trial being conducted by Sherer, which is looking at neurotrophic factors, and particularly the neurotrophic factor nurturin. Neurotrophic factors function to promote the survival of brain cells, and in preclinical models, neurotrophic factors have been shown to be neurorestorative, which means they could bring sick cells that are affected by the disease back online to function more properly. So that we're very anxiously awaiting those results. The trial, all the subjects have been treated, and they're just waiting to complete the evaluation of the patients that are involved in the project. And we continue to get more data from the Parkinson's Progression Markers Initiative, which should complete the baseline time zero newly diagnosed data set to really try to optimize some diagnostic tests for Parkinson's. There are a number of trials that will also be launched in 2013. There's a novel collaboration between the Fox Foundation and Sanofi on a treatment for cognitive dysfunction in Parkinson's. A trial that the Fox Foundation is working on with researchers in both the United States and Germany to test nicotine patches as a potential disease-modifying therapy. This builds off some interesting epidemiological data which suggests that smoking can actually reduce your risk of Parkinson's. Nicotine itself actually functions in the motor system of the brain, so there's both epidemiological and scientific reasons to support this as a potential therapy. This will be the first trial really looking at this for disease modification in Parkinson's. And we're also expanding efforts to try to identify individuals prior to the diagnosis of Parkinson's, to identify people at risk for Parkinson's disease, and that will get launched as well in 2013. So just as a summary now, looking forward, so we started where we were in 2000, where are we today, and where could we be 5 years from now with proper funding and if all scientific milestones are met. There's a very robust pipeline of potential therapies in Parkinson's that are moving through clinical development and have a lot of potential to being in patients' hands in the short term. Again, caveating with all the proper funding and scientific milestones. So it's a very exciting time in Parkinson's research, and we hope that we'll be able to build on some of the results in 2012 into 2013. So now we're going to open it up for the question and answer session. I hope I covered some of the questions during the overview presentation. And just as a reminder, you can submit these through the system online, and we will do our best to answer as many of them as we can during the call, and then we can post some answers afterwards. One of the first questions that came in had to do with impulse control disorders. I'm actually going to turn to Maurizio to start the answer on this. What are they, where do they come from, and is anything being done to develop treatment?
Well, thank you, Todd. Hi, everybody. Thanks for the opportunity to be with you today. So impulse controls are mainly just to be brief on that, and I will be happy to take more questions through the internet. But they're mainly related to the therapy. Most of the people that they experience this kind of like a compliance are taking a specific category of dopaminergic therapy is a dopamine agonist. That doesn't mean that only that specific category of drugs might develop, might actually lead to the development of impulse controls, but just like it seems to be more predominant with that specific category of drugs as to opposed to dopamine itself or levodopa, carbidopa, as you well know. What are those? There are several possible symptoms when it comes to impulse controls. I mean, we talk about an increase of hunger, for example, you need to go out and get as much food as you can, or go and there's pathological shopping. I mean, one of the most reported thing is the pathological gambling, for example. But there's also hypersexuality. That's something that we should not forget. So all of those things are determined by an increase of the dopaminergic system. And it's funny because we need it to increase the dopaminergic response. For Parkinson's, that because we know that the motor part of Parkinson's disease is related to a lack of dopamine, but dopamine doesn't work only on the motor part of the disease. It's just in the brain works in 3 to 4 different other pathways, and one of those is about rewarding. So if we increase dopamine, we increase the seek for rewarding, and that sometimes in the pathological level leads to increasing that feeling of feeling satisfied by buying new clothes when you even maybe don't need them, or buy new items or food, and the whole things that I said before. It's a very simple thing to treat. It's very annoying, so it's something that we understand. We need to work on that. So don't feel afraid to communicate that with your physician if you even suspect that and you're taking a dopaminergic therapy. So be free to talk about it. It's very easy to deal with. Most of the times, unfortunately, requires an adjustment of the therapy that you're taking. We understand that might impair, the balance for a moment, but after a couple of weeks of readjusting the therapy, everything gets back to normality. There are other specific therapies that they sort of counteract the effect of that, of the dopaminergic therapy, but you don't want to go that way because it would be like taking something and taking something else is blocking the effect of the first one. So I think it's just a matter of adjusting that. There is no specific research on that because we realize that is more a side effect of the therapy and is not completely related to the pathology of the disease. So we are definitely working on improving what we have as a therapy more than actually doing an active research of understanding why those symptoms come out, because we know that those symptoms are just related to an increased stimulation of specific receptors when it comes to the dopamine system. So I hope I actually answer that first question, and happy to take more.
Great. So one of the other questions was actually a very detailed question, and I definitely want to answer it because it's a good point. So someone raised, what about the new impacts dopamine delivery approach? And I just want to bring this one up because I think it should have been mentioned more explicitly in my presentation. So there has been a lot of work on improving levodopa delivery, and if you see here, I have this highlighted under the symptomatic treatments. What impacts is developing is a levodopa cinnamate that is in the way it's handled in the body is longer lasting with each sort of pill that's taken, and it has been shown to increase the on time without causing dyskinesias or troublesome dyskinesias, or one other way of saying it is decreasing the off time that people have during the course of a day. And this is up for FDA approval. We thought we were going to actually get the FDA response this fall. It's been delayed till, I believe, January or February, but we should know very soon whether that is a new drug that will be available in sort of the arsenal of potential treatments, particularly in balancing the different ways to increase dopamine in the brain. So that is a very promising therapy. The foundation had a small project with the company to work on this, and I do think it could have a pretty good impact in terms of the ability to treat the disease in the short term. So some of these examples that we are raising could be, approved within the next 18 months from the FDA. So it's a good one to highlight in terms of that line on the levodopa delivery. Another question, and I'm going to turn back to Maurizio on this because he also is an epidemiologist. So the question is, what have been the advances on the environmental causes of Parkinson's? We did talk a lot about, some on the genetics in this call, but What do we know about the environment?
So, as you well know, in the current state of the interpretation of how Parkinson comes is a combination of a genetic predisposition as well as possible environmental toxic factors. The challenge is that to date, we still have some challenges in defining which of those factors are. There is a current research that, and actually we as a foundation, we are funding and supporting part of that research that works on specific chemicals, and more in detail is like chlorines and other constituents of detergents and something like that. But just to let you know, I always, when I was seeing patients, I always said that, it's like if we only related to one specific toxic, we will see way more, the effects of that, and we'll be seeing many more patients. So despite the fact that we can maybe work and find out a specific toxic or environmental cause, is always, we still need to understand whether it's like the exposure, the duration of the exposure, or the moment that someone has been exposed, and, that genetic predisposition. So the same way that no Parkinson is the same between people, everybody has a different presentation of their own disease, there's also a different effect of those toxic factors on our body. Some of those they have the ability to detoxifying faster, some others they don't, and again is part of their genetic predisposition. So what I can tell you is that there is still research going on there. We definitely didn't completely forgot about it, but in the same way as Todd mentioned before, the role of genetics, so we know that genetics helped us understand the pathophysiology of Parkinson. So what, a gene is causing a specific alteration in a protein, and then we can actually do the same way with the toxic environment and say, environmental toxic, and say that those are altering specific proteins, so they help us understand which is the pathway behind Parkinson's disease. So I actually don't have a very a thorough and comprehensive answer of what are those toxins right now, but the research studied that way. They started with specific pesticides. We exclude several categories of pesticides, so the ones that we are currently using right now are safe. But definitely from that moment on, a lot of chemists are now interested into finding molecules that might interact with those proteins that we said before, and together with the genetic learning that we are accumulating these years, we're trying to define a more comprehensive and better pathway. So I hope again I answered, but it's a complex question, I guess.
There was a question related to the gene therapy trials, so I'll answer this one. Specifically, the question is, in the gene implant therapy trials, are there more than one gene being studied? And the answer is yes. So I highlighted in the presentation a project that Sherer is doing, which is a gene therapy trial for a neurotrophic factor called nurturin. The way that gene therapy works, so it's overcoming a challenge. So a challenge that the brain has, or the body has, what we have in developing a neurological treatment is that the body has something called the blood-brain barrier, which prevents certain molecules, particularly larger molecules, certain potential therapies from getting into the brain. The blood-brain barrier is normally a very good thing in that it also prevents pathogens or other toxins from getting in the brain. But it becomes a real challenge for getting drugs or therapies into the brain. So what gene therapy does, it does involve a surgical procedure into the brain, but then allows the brain itself to generate the therapy. So in the case of Sherer, they use genetic techniques to using the surgery to then promote the ability of the brain itself to develop that neurotrophic factor. So there are other gene therapy projects going on. There's another neurotrophic factor therapy that's being conducted by the NIH, which is using GDNF as the neurotrophic factor, which is closely related to nurturin, which Sherer is doing. There's another trial that the Fox Foundation is working on with a researcher in San Francisco. This one will probably start, I would guess, towards the end of 2013, maybe 2014, which is using a gene that helps the brain make more dopamine. So that one is ongoing. And there was a trial that was conducted in Europe which also used genes that allowed the brain to make more dopamine. So there's really been two aspects of this. One is optimizing the techniques for delivering those genes to the brain, and then there have been an array of different specific therapies, specific genes that are being tested right now up to the stage in clinical testing. So another question related to exendin-4 as a potential therapy for Parkinson's, and Maurizio has actually followed this pretty closely, so I'll turn it over to him.
All right, so exendin-4 is a target that, so I would actually, let me do a little a step back. There is a category of drugs called GLP-1 antagonists, and those are agonists. They're all drugs that have been developed for diabetes, for this, the second form of diabetes type 2. So how we got those drugs working for Parkinson's, or why we are interested in that, because they, for pure anecdotal evidence, they found out in animal models of Parkinson's disease by administering drugs such as exendin, exenatide, which is working on exendin-4 and is GLP-1 agonist. They were reversing a little bit of the effect of Parkinson's disease on those animal models. So, as usual, that is sort of an epiphany kind of thing. People find something, that is worked for another disease, and they try to move it, and we call it reposition it into Parkinson's, which is something very interesting because it will accelerate, the process of testing that drug in Parkinson's, just because it's been already proved safe in another category of individuals, like the diabetes people. So that drug is now being tested in human beings. The good thing is that, again, the same story as those drugs working on the glutamate for dyskinesia. Even in this case, we have 3 different companies that are interested, in working on those drugs for Parkinson's. So they makes us feel that GLP-1 agonists are indeed a very good category of drugs that we are very interested into, and we want to see what is going to happen. That we are a little bit early in the clinical testing, so we cannot. So I think we will wait. At least 2 to 3 years before knowing if that is really effective in human beings, especially in individuals with Parkinson's disease. But it's interesting and is promising. And another thing that I want to do, I want to just mention, is that it's funny how we are learning from different diseases, including from the Alzheimer's field, for example, and the connection with diabetes, and maybe between diabetes and the immune system, and there's a lot of intricate and complex pathways that are now getting together and trying to explain, or make a little bit more clarity around Parkinson's disease. So we are learning from those fields, we're learning from those diseases as well, and we since we are learning now, and they were doing that before, we are accelerating that, and we try to make as many mistakes as possible, leveraging from that work, and moving these things as fast as possible towards the clinical stage. So there are a number of questions related to stem cells, and
I think one of the reasons is when I posted what was really exciting in 2000, we really highlighted the potential of stem cells. And I want to just explain sort of where we've gone in stem cells in the last 12 years and where the future may be for stem cells in terms of developing therapies. So the concept behind stem cell therapy for Parkinson's disease is to utilize the stem cell to develop and engineer the stem cell into the cells that are lost into the disease. So in Parkinson's specifically, to take a stem cell and make it into a dopamine-producing cell and then transplant that cell into the brain of a patient with the disease to replace the cells that were lost in the disease. So that sounds like a very straightforward concept, and that's really where the whole excitement and research started. What we found through the research is that embryonic stem cells remain the best source of stem cells for the capability of being made into dopamine-producing cells, and there have been some very sophisticated techniques developed in culture dishes to make that conversion from an embryonic stem cell into a large number of dopamine-producing cells. One of the main challenges that has occurred, however, is once those cells are transplanted into a brain, into the environment of the brain, they seem to revert back to stem cell phenotype or stem cell characteristics. So what that means is they lose that engineering of ability to produce dopamine, or they are not as efficient at doing that as they were in the culture dish. So that has become a major research challenge that has been a focus of a lot of research. And recently there has been some advances on this to improve the ability of those cells to survive and continue to produce dopamine in the brains, and this is in preclinical studies. So there is a program in New York that is continuing to work on this and push this forward, trying to now really address a lot of the potential safety issues and address issues of generating enough cells to really be viable for therapeutic potential, given the amount of cells you'd have to transplant into a brain. I do think there's an important caveat in what we've learned, though, with stem cells is that really the potential outcome of this is really to target treatments that are responding to dopamine-based treatments, because it's another way of getting dopamine into the brain. So I still think it has some potential to be a significant therapeutic approach, but I do not think that it has the capability of the full spectrum of solving the whole issue of Parkinson's. And that's for one reason, I think, combination of technological challenges and that potential, why it has been more de-emphasized by the field than it was 12 years ago. Now, one last point on stem cells, though, is in the last couple of years there was a major advance in the ability to make stem cells out of somebody's skin cell. So to take the skin cell from an adult and convert that back into a stem cell. Why that has been very exciting broadly is that you could envision sort of doing a self-transplant, because you're going to get a cell from someone with a disease and then use that cell to transplant back, so you can get rid of some of those challenges around transplant rejection. But I think more importantly for Parkinson's disease is now we have the capability of developing models of the disease that could be used for testing new hypotheses and new therapies. So we have a challenge in Parkinson's disease if you compare to a disease like cancer, in that in cancer you can actually remove the diseased tissue and study that tissue. In Parkinson's disease, the diseased tissue is predominantly the brain, so we don't really have a lot of access to that for a few, laboratory studies. But with this new technology, you can develop cells from people with Parkinson's. We'll have the genetic makeup of that person with Parkinson's and now study that for both biology and drug development. And that is where the field has also gone with stem cells. So I want to just turn now to Maurizio. I think the general question is, we talked a lot in this presentation on drug treatment or biological, surgical treatments. What about things like diet and exercise that people could do today?
Right. So yes, we got several questions about diet, and I will like to just try to summarize a couple of things. So first of all, in general, for anybody, there is not a specific diet recommended, meaning that you can eat everything. It's just like there is no restriction on the kind of food you can get. So just get everything in a good balanced way. One thing that is important to know, though, and not everybody knows it, is that most of the treatment for Parkinson's disease, especially if you go on Sinemet, or levodopa carbidopa, levodopa is an amino acid. An amino acid is a protein. So let me do a step back. I mean, the levodopa is absorbed not in the stomach, but in the first part of the intestine. So if we get a very heavy, fatty meal, the time that the meal goes from the stomach to the intestine, it takes way longer than just eating carbs or a salad or something like that. So the drug, before moving from the stomach getting into the intestine, takes longer, and in that time is destroyed by the stomach. So you understand, you appreciate the fact that the absorption of levodopa is reduced by a very heavy, fatty kind of meal. The second thing is that levodopa is, as I said, an amino acid, so it's like as a protein. So if your meal is mainly constituted by meat, but proteins, cheese, meat, something like that, that also compete with the absorption of levodopa. So what is my recommendation in general, and I think it should be a good practice, is that during the day we should be very careful the moment that we take levodopa, carbidopa, and most likely I would say a half an hour before lunch or before a meal, so that you can allow that pill to go through the stomach and get to the intestine and being absorbed before you eat, or a full hour after a meal. So you allow your stomach to empty, and start digesting your food, and then the pill goes down, to the intestine. If isn't better, in order to avoid those off and on times, these fluctuations that are common in the late stage of Parkinson's disease, one of my recommendations would be try to avoid meats and cheese during lunchtime or during the day. So most of the people will not take levodopa carbidopa before going to sleep. So why don't moving those specific foods at the end of the day? It's it's like a sport kind of diet, so where you have carbs and vegetables and something like that during the day at lunchtime, and then you prefer to have fish, meats, and cheese at the end of the day. So that's just a recommendation for those who are experiencing those motor fluctuations. There was a specific question about grapefruit. So grapefruit is accelerating the metabolism of several compounds, but not a levodopa. So don't worry about that. Levodopa doesn't get metabolized by the liver. It goes directly, and the reason why we use levodopa carbidopa is because it gets directly into the brain, is absorbed by the dopaminergic neurons and transformed into dopamine. So no effect, no combination. There is no known specific interaction with other drugs. So be safe on that. It's not a problem. So there is no specific food recommendation. Just be careful of fatty or heavy meals that will delay the transition of the food through the intestine, and as well as protein. That is the only recommendation that I give on diet. For exercise, I will be even shorter. Exercise is good, period. It's there is no specific, and we did a lot of research. So to the questions that were is the foundation funding, sponsoring, working, supporting research on exercise? Yes. We did a lot in the past. We are still doing something now. But let me tell you, there is no specific exercise. There's not the magic exercise. A lot of people are finding benefit in martial arts, some others in swimming, some others in biking. So there is no specific exercise. Whatever works for you is the right one. 2 recommendations. First of all, get something that you can do constantly. So don't overdo the first day because you just oh, right, now the new resolution, I'm going to go and exercise for an hour, and then you're going to get dead, because it's going to be too much. No, so to do something, but constantly. Not every day. It's fine, maybe twice, 3 times a week, but keep your body in movement. That is very important. The joints are getting sick if we don't move them. So we need to move them. We need to keep the exercise going. The second thing I think I forgot. I just lost my but it was like, yeah, no, I don't know. I forgot what I wanted to say. But the point is, there is no magic exercise. Just keep on doing that. And there is a good evidence also when it comes to biological factors that is not only a mechanical thing, but is producing some specific transmitter in the brain that are not only giving you a sense of of wellness, but it seems to be neuroprotective. Again, that's a theory. There is a lot of work going on there. Those neuroprotectants are increasing in the brain after exercise. They are staying there for quite a long amount of time, but they will eventually go down. So that's the reason why constancy and keeping on doing exercise is important. So I will close here for the exercise thing. We do think it's a priority, but might not be a priority like a research priority right now, because we confirm that it is important and has to be done.
Okay, thanks, Maurizio. We are approaching the end of the hour, and I know there's still a lot of questions we're probably not going to get to. So I just wanted to start by first thanking everybody for participating and for all the questions. What we are going to do is do our best. This presentation will be posted, including the recording, so you can share this with as many people as you want, as many people as you think would be interested. We will also try to provide some answers to some of the questions that have been posted that we didn't get a chance to address. Particularly, there are some that focus on some very recent research results that I think we'd like to explore further. So we'll do the best we can to answer those. A couple of other things, we would really appreciate if people would complete the survey we'll send around to give feedback on this new model of these calls, particularly integrating the video to help us as we're building. We're starting now to plan for next year's, so we're also very interested in feedback on specific topics people would like us to cover in the series next year as well. And one last point is we mentioned the important role that all of us can play, both people with Parkinson's disease or people without Parkinson's, in helping to accelerate the research by getting involved in the research. And I want to just remind people about Fox Trial Finder and working to educate yourself about what the opportunities are to get involved in research at your level of comfort. So I want to, again, thank everybody for participating, for the great questions and highly engaged interaction. And please look for the information on the website, and we will provide more answers to some of those questions as best we can.